So imagine paying, I don't know, $3,000 to have your own blood drawn, spun down in a centrifuge, and then carefully injected into your torn knee tendon.
Yeah.
Or, maybe your receding hairline.
Right. It's a huge investment.
Exactly. You are investing in cutting-edge regenerative medicine. But then you realize that the single glass of wine you had at dinner last night just physically warped your cells, like vaporizing up to 40% of your treatment's effectiveness before the needle even touches your skin.
Yeah, it's wild to think about.
It really is.
Yeah.
So today on our deep dive, we are tackling this massive invisible problem. We're looking at how systemic alcohol dependence, or honestly even just casual drinking, completely dismantles your body's regenerative abilities at a microscopic level.
It is a phenomenal hidden mechanism. We are looking at a collision between, everyday habits and highly complex cellular architecture.
Because our source stack today is this fascinating mix. We've got practical clinical advisories from orthopedic and rejuvenation clinics sitting right next to these incredibly dense biochemical studies and proteomic reports.
Right. And merging those two worlds is where the real story is. We're going to explore the systemic impact on platelet membrane fluidity, the disruption of growth factor release kinetics, and why something as ubiquitous as ethanol can turn a powerful healing procedure into a biological dead end.
Okay, let's unpack this, because whether you are an elite athlete trying to get back on the field or, someone looking into skin rejuvenation, you are trusting your own biology to do the heavy lifting. You're paying for your own blood.
Absolutely. You really are.
Platelet-rich plasma, or PRP therapy, relies entirely on your body's innate materials. But we are going to explore exactly why having a few drinks doesn't just dehydrate you, it actually physically sabotages that biological investment.
Yeah. And before we look at the cellular destruction, we first need to establish the baseline, right? We need to understand what a healthy platelet is actually supposed to do during one of these PRP treatments.
Right, for the listener who maybe just knows the basics, it's drawing a small amount of blood, spinning it to concentrate the platelets, and injecting it. But why are platelets the star of the show here?
Well, to appreciate the destruction, you really have to appreciate the design. Platelets are unique in human biology because they are anuclear. They do not have a nucleus.
Okay.
So instead of acting like traditional cells that divide and replicate, I like to think of platelets as these incredibly efficient microscopic cargo ships.
Yeah.
And they're constantly circulating in your bloodstream.
And the cargo they are carrying is what we are actually paying for at the clinic.
Precisely. They are loaded with highly specialized growth factors. When we talk about regenerative medicine, we are primarily talking about platelet-derived growth factor, or PDGF, and vascular endothelial growth factor, VEGF.
Right, the heavy hitter.
Exactly.
Right.
These aren't just generic nutrients. These are the master signaling molecules that literally dictate tissue repair. When a platelet encounters an injury, it releases these factors to jumpstart the formation of new blood vessels, suppress harmful inflammation, and trigger this massive collagen production.
So they act like a biological signal flare, basically telling the body to rush all available resources to this exact coordinate and start building new tissue.
If we connect this to the bigger picture, the entire premise of the therapy relies on those cells being robust, structurally sound, and fully loaded with their cargo. They need to be ready to release it in a localized, explosive burst right where the clinician injects them.
But the conflict here is pretty stark. The clinical studies we looked at show a staggering 20 to 40% drop in the effectiveness of these treatments, linked directly to alcohol consumption.
Yeah, up to 40%. It's huge.
And this isn't just about showing up to your appointment feeling a little dehydrated. The analogy I kept thinking of is that getting PRP is like hiring a really expensive construction crew to fix your house.
That's a great way to look at it.
Right. But drinking beforehand is like the crew showing up hungover. The needle goes in either way, but the cells you are delivering to the job site are basically half asleep.
Yeah, to understand why they're half asleep, we have to zoom all the way in. We need to look past the fluid of the blood and examine the actual physical architecture of the platelet's outer shell, the cell membrane itself.
Let's definitely break that down, because I think people assume alcohol just floats around in the blood water, just changing our mood. But it is actually changing the physical shape of the cells.
It really is. The thermodynamics of the cell membrane are incredibly delicate. A membrane is primarily a phospholipid bilayer.
Right.
You can visualize it as this fluid, flexible wall made of fatty lipids and water-soluble components. It insulates the intricate machinery inside the cell from the absolute chaos outside.
And ethanol just messes with that.
Completely. Ethanol, which is the type of alcohol we consume, is what we call an amphiphilic molecule, meaning it has a water-loving end and a fat-loving end.
Oh, interesting. Which means it doesn't just float in the water of the bloodstream. It has the chemical passport to basically go wherever it wants.
And it uses that passport to physically wedge itself right into the lipid-water interface of this protective cell wall. It literally partitions into it.
Just shoves its way in.
Exactly. By forming hydrogen bonds with the lipid molecules of the membrane. So it doesn't just passively bump against the cell, it chemically integrates into the structural foundation.
So think of the cell membrane like a sturdy brick wall held together by thick mortar. The alcohol isn't just rain washing over the wall. It acts as a solvent that dissolves the mortar. The bricks actually start to slip.
Yes. And that slipping is a deeply measurable phenomenon. Higher ethanol concentrations cause a massive physical reduction in the thickness of the lipid membrane, up to a 30% reduction.
Oh, wait. The cell membrane physically shrinks by a third.
Yes. In cellular thermodynamics, this state is called the interdigitated phase. It induces highly abnormal membrane fluidity.
That sounds crazy. Doesn't that completely destroy the proteins and receptors embedded in it?
That is exactly the problem. And it triggers a cascading failure called hydrophobic mismatch.
Hydrophobic mismatch. Okay, what does that mean in plain English?
So integral membrane proteins are anchored inside the cell wall. Crucial parts of these proteins are hydrophobic, meaning they chemically repel water. By design, they sit perfectly shielded inside the fatty, water-free core of that thick lipid membrane.
Wait, I see where this is going. If they are hydrophobic, like terrified of water, and suddenly the alcohol thins out their protective fatty coating by 30%.
They are suddenly exposed to the watery environment of the bloodstream.
They must completely panic.
In a biochemical sense, yeah, they panic. To avoid the surrounding fluid, these newly exposed membrane proteins frantically try to hide. They start clumping together, aggregating, or completely changing their conformation just to shield their hydrophobic amino acids from the water.
That is incredible. So for you listening, when you have a couple of drinks, alcohol isn't just giving you a buzz. It is fundamentally, microscopically warping the physical shape and thickness of the cells circulating in your blood.
Yeah, they're twisting and clumping up. And what's fascinating here is how this external physical warping triggers a massive premature chemical reaction inside the platelet.
Because the outside is ruined, the inside goes haywire.
Exactly. The signaling pathways housed inside that membrane just completely break down.
This brings us to a concept in the sources called the unified theory of alcohol and platelets. I found this part deeply counterintuitive, because alcohol actually does two completely opposite things to a platelet at the same time.
Right. It has both inhibitory and activating effects. It's a real paradox.
Yeah, walk us through that.
So to decode it, let's look at the internal brake system of a healthy platelet. Inside the cell, there's a secondary messenger molecule called cyclic AMP.
Okay, cyclic AMP.
Its primary job is to act as a chemical brake. It keeps the platelet calm and stable while it circulates. You absolutely do not want a platelet activating and dumping its growth factors while it is just floating through normal, healthy blood vessels.
Right, that would be a disaster. But alcohol cuts that brake line, doesn't it?
It forces the brake line to fail. Ethanol transiently lowers the internal levels of that cyclic AMP messenger molecule. By removing that biological restraint, the alcohol actually partially activates the platelets.
Here's where it gets really interesting. Because they are partially activated early, long before they ever reach an injury site, they undergo partial degranulation.
Yes, they start leaking their precious cargo.
Right into your general circulation. So by the time you sit in the clinician's chair, have your blood drawn, and the centrifuge spins it down, those platelets are suffering from profound platelet exhaustion.
They have already fired off their biological flares.
So the construction crew has already dropped their tools before they even arrived at the job site.
It's a perfect analogy.
Yeah.
And the downstream chemical failures of these exhausted platelets are just catastrophic for a regenerative procedure.
What else goes wrong chemically?
Well, because of that hydrophobic mismatch we talked about, the platelet has an impaired ability to mobilize arachidonic acid, which leads to severely reduced generation of thromboxane A2, or TXA2.
And TXA2 is what they need to create their own biological glue, right?
Exactly. So the anchoring system goes completely offline. On top of that, there is an inhibition of the expression of a specific surface receptor called alpha-2-beta-3.
Okay, that's a mouthful. What does that receptor do?
It's crucial for fibrinogen binding. Without it, The platelet's ability to aggregate drops within minutes of a drink, and it can stay blunted for hours.
Which brings it all back to the listener and the core mechanism of PRP.
Huh.
During the therapy, you need an active, explosive release of growth factors directly at the injury site.
Right at the knee or the scalp.
But because of this membrane exhaustion, the release kinetics are completely ruined.
Yeah.
You are paying thousands of dollars for exhausted, deformed cells that can't stick together.
And unfortunately, it gets worse. Because the destruction doesn't stay confined to the test tube or the centrifuge, we also have to evaluate the systemic environment that these exhausted platelets are being injected back into.
Right, the rest of your body.
Exactly. The biological ecosystem itself becomes deeply toxic to the healing process. Let's look at the hormonal disruption first.
The numbers on this were shocking.
They really are. Processing ethanol triggers the body's stress response. Within hours of consumption, alcohol can cause cortisol levels to spike by up to 152%.
152%. That is a massive systemic biological panic attack.
And cortisol is highly catabolic. It breaks things down. That spike actively suppresses the release of growth hormone, which is vital for tissue regeneration, and it simultaneously reduces testosterone.
So your body is being chemically instructed to stop building new tissue, right at the exact moment you are injecting cells designed to build new tissue.
It's completely counterproductive. And then we factor in the inflammatory imbalance.
Right, because PRP relies on a very localized, carefully controlled inflammatory response to signal the repair cascade, right?
Yes, controlled being the key word. But alcohol floods the entire body with pro-inflammatory cytokines, like TNF-alpha and IL-6.
It's like needing a small, controlled campfire to cook a meal, and someone just drops a bomb on the entire forest. The body's immune system just doesn't know where to focus.
Exactly. And while those systemic wildfires are burning, alcohol simultaneously depletes the body's stores of glutathione, which is our primary antioxidant.
So you've triggered massive inflammation and disarmed your body's main defense at the same time.
Which perfectly explains the clinical data we see on muscle repair. When the cellular environment is that toxic, muscle protein synthesis drops by up to 30%.
Wow. 30%.
And on top of that, alcohol suppresses vasopressin, acting as a powerful diuretic. It forces aggressive dehydration.
So what does this all mean? If we summarize this for you, doing a PRP treatment with alcohol in your system is basically like planting highly expensive seeds in dry, toxic soil, and then wondering why nothing grows.
Yeah, you've ruined the seed and poisoned the soil.
Which brings us to the most actionable part of this deep dive. If your cell membranes shrink, the platelets get exhausted, and the soil turns toxic, how long do you actually need to abstain to protect your PRP investment?
Well, we have to look at the life cycle of the cells. A standard platelet circulates in the human bloodstream for roughly 7 to 10 days.
Okay, 7 to 10 days.
Based on that, regenerative clinics typically demand an absolute minimum of 72 hours of strict, complete abstinence prior to the blood draw.
72 hours. 3 full days of zero ethanol.
This raises an important question, though. Why 72 hours specifically?
Right. Why not just 24 hours to let it get out of your system?
Because it's not just about filtering the alcohol out. You need that 72-hour window to allow your bone marrow to generate a completely new batch of fresh, unexposed platelets.
I see.
You need a critical mass of healthy cells that haven't suffered from that ethanol-induced interdigitation and thinning we talked about.
They need to be entirely free from those panicked, clumped-up surface proteins.
Exactly. The 72-hour window ensures the centrifuge is concentrating healthy, fully loaded cargo ships. But the timeline definitely doesn't stop once the procedure is over.
Right, because the 48 to 72 hours after the injection are just as vital.
Arguably more vital. That post-injection window is when the growth factors are actively binding to receptors and signaling tissue repair. If you drink then, you instantly spike that cortisol, flood the system with inflammatory cytokines, and ruin the healing cascade.
You're tearing down the scaffolding while the crew's trying to lay the bricks. And there's a surprising caveat in the sources regarding chronic heavy drinkers that really caught my eye.
The hyperaggregability rebound.
Yes, people who drink heavily and then completely withdraw, their platelets actually get super sticky.
It is a delicate biological paradox. For someone who drinks chronically, their body builds up countermeasures to fight the constant inhibitory effects of the alcohol.
So their internal brake system gets used to the alcohol.
Right. And when they suddenly withdraw completely, those countermeasures are still active, but the alcohol is gone. The result is a massive rebound effect. Usually 1 to 2 weeks after they stop drinking, their platelets become hyperaggregable.
So instead of being asleep, the cargo ships are hyper-agitated and crashing into each other.
Exactly. And this overreactivity parallels severe simultaneous changes in the patient's blood plasma, specifically huge fluctuations in the ratio of LDL to HDL cholesterol.
So the entire lipid environment just goes through this turbulent adjustment phase.
It proves that returning the body to a natural, pristine baseline is a highly complex, time-consuming biological process.
That is just wild. So let's bring it all together for you listening. Avoiding a drink around your PRP session isn't just generic wellness advice to, help you feel better.
No, it's a critical, non-negotiable requirement.
Exactly. It's required to prevent ethanol from physically thinning your cell membranes, exhausting your platelet signaling, and basically poisoning the hormonal environment required for healing.
You simply cannot out-engineer a compromised cellular membrane.
Which leaves you with a pretty provocative final thought to mull over. If a simple chemical like ethanol can physically partition into your lipid bilayer and alter your cell membrane thickness by up to 30%.
It's a scary thought.
It really is. How might other daily, seemingly harmless dietary habits, or even just stress-induced chemicals, be secretly altering the physical shape and thickness of our cells right now, without us ever even knowing?
It definitely challenges everything we think we know about our daily routines.
Absolutely. Something to explore on your own. Until next time, thanks for joining us on this deep dive.